Hello, my name is Miranda Meelan. I'm a PhD student at UC Santa Cruz in Ingrid Parker's lab. Today, I'm going to tell you about my research on how Detrichia graveolens has responded to population spread in its European native range and while invading California. Globally, many species are expanding ranges, either in response to climate change in their native range or after an introduction to a new environment. In order to understand whether species will track climate or colonize new areas, it is critical to understand the drivers of population spread. The ability to accurately predict the spread and impact of invasive species is necessary for effective risk management in natural and agricultural systems. Due to the complexity of the process of invasion, it is quite challenging to develop methods of forecasting population spread. The ecology of population spread has been researched for a long time and predicts that spread reaches a constant asymptotic velocity over time. Evolution may speed up this process, greatly complicating range expansion forecasts. With multiple experiments showing how invading populations can evolve within a few generations, evolution, in addition to ecology, plays an important role in understanding the drivers of invasion. Ecological theory shows that in homogeneous landscapes, the spread velocity of a population is driven by the success of those at the leading edge of the expansion. Those front runners produce the offspring that propel the population forward. Therefore, traits of high dispersal and fecundity drive spread, and evolutionary theory predicts that those traits are thereby under strong selection on the leading edge. With evolution in the equation, now, the rate of spread may accelerate. We must also consider that as populations spread to new areas, they encounter new conditions, so local climate is another source of adaptation. For this presentation, I will be focusing primarily on dispersal. For a wind-dispersed plant, we predicted that those individuals at the leading edge of range expansion or invasion would have evolved traits that increase fecundity, such as an increase in the number of seeds and an increase in the number of seed heads. We also expected the evolution of traits that promote dispersal ability, such as lighter seeds and taller plants. We have been studying differentiation in dispersal and reproductive rates in a species that is currently expanding its native range and invading a new continent. Detrichia graveolens, also known as stinkwort, is a small herbaceous plant with aromatic and sticky foliage. The plant is harmful if ingested by livestock and can cause contact dermatitis in some humans. It is a late-season, fall-flowering plant with yellow radiate flowers that produce wind-dispersed fruits. Commonly found in disturbed soils and along roadways, Detrichia is native to the Mediterranean basin in Europe and has successfully invaded other continents, including Australia and North America. Here in red, you can see the native range within Europe. We collected from populations in 3 regions in Europe: from southern France, from central France on the northern periphery of the historical range, and from the Netherlands, where the species has recently expanded. We know from previous genetic work that central France was the origin for these populations in the Netherlands. In California, Detrichia was first introduced in 1984, right here, in the San Francisco Bay Area. In the past 30 years, it has spread eastward and upward into the Sierra foothills. We collected populations from 2 regions, from the original core of the introduced range and along the spreading edge. Seeds from 46 populations and up to 13 replicates per population were grown in a blocked design at the UCSC greenhouses. We measured several plant traits that are expected to be under selection during range expansion because they promote population spread. These include number of seeds per seed head and the number of seed heads per plant, which affect fecundity, and seed weight and plant height, which affect dispersal. Let's look at the fecundity traits. Recall that for a wind-dispersed plant, we predicted that those individuals at the edge would have evolved traits that increase fecundity, leading to an increase in total seeds per plant. We expected evolution for higher fecundity in spreading edge populations, such as the Netherlands, and in the California edge population. However, edge populations produced fewer seeds. One possible mechanism here is that reduced genetic diversity during spread and translocation led to the expression of inbreeding depression at the seed production stage. Consistent with this explanation, we found that the reduction in total seed number was driven entirely by reduced seed number per head, possibly suggesting partial reproductive failure. Okay, so moving to our measurements on dispersal ability. Recall that for a wind-dispersed plant, we expected the evolution of traits that promote population spread, such as lighter seeds and taller plants. Although we expected to see lighter seeds as the populations moved north in Europe, we don't see any strong pattern in seed weight. So let's look at plant height. Well, contrary to what we expected, as the plant spread further away from the core region in southern France to new areas, plant height did not increase. It decreased. And over in California, we see that plants are not taller than southern France, nor is there any significant change in the height between the core and edge populations in California. So we see strong differentiation in plant height, but not expressing a signature of selection for increased dispersal. But remember that as populations spread, they may also encounter and adapt to new environmental conditions. So, if plant height grouped by region didn't show our prediction of taller plants in edge populations, we thought climate may be a driving factor. Here is Detrichia's native range, which is primarily confined to the Mediterranean basin. Our populations in central France are right on the periphery of the range, and populations in the Netherlands are far north of this boundary. Starting from the earliest work in plant evolutionary ecology, we know that plant height responds to latitude and elevation, with colder climates leading to short stature. So how does Detrichia respond? When we graph our individual populations by minimum temperature, we see a strong signature of smaller plants in cooler sites. When we add California populations onto this graph, we see the California core populations are very similar to the plants from the similar Mediterranean climate of southern France. Here, in bright red, you can see the populations that have very recently colonized the foothills of the Sierra Nevada mountains. We expect these populations to evolve shorter stature, but we have not seen that, at least not yet. We saw that traits in Detrichia graveolens did not show a response to selection for rapid spread. We predicted edge populations to have higher seed production, but found they have lower seed production. We expected seed weight to be lighter in edge populations, but we saw no difference. We also expected edge populations to produce taller plants, but they were shorter in spreading populations. Finally, we see evidence for other selective forces, such as climate. We see shorter plants from regions with colder temperatures, but no evidence in California for adaptation to high elevation. My co-authors and I would like to acknowledge those that have assisted in the collection and production of this research, our collaborators and our funders, the USDA NIFA program, the Department of Ecology and Evolutionary Biology at UC Santa Cruz, and the Swiss National Science Foundation. Thank you.